think I would have to say the interactions with the students. The thing I love most about being in a classroom is when students are engaged. When I kind of pose a question to the class and I see a lot of students interested in participating, and then we can start a discussion on things. So I think that interaction is what I really love most about teaching. And the other thing is, and I also really love it when students ask me questions that I've never thought of, right? This kind of challenges me, and sometimes it makes me see things from a different perspective. So the motivation for the work that we do is because of sustainability and environmental issues with the current petrochemical routes of production of fuels and chemicals, that we need to seek alternative routes. So in my lab, we're focused on bio-based routes of production. Now, in the development of bio-based processes, mainly in the use of bio-based catalyst for chemical conversion, there are a lot of existing challenges in the development of these biocatalysts, mainly because, well, we don't know everything about biological systems. We don't understand fully how they work. And if we don't fully understand how they work, then we can't optimize them as well as we possibly can. So the little bit of a background, the way that biological systems work, right? Cellular properties or functions are encoded by their DNA sequences. And because DNA sequences can change, right? Mutations, right? So a change in the DNA sequence is called a mutation. Because mutations can occur, we can actually modify cellular properties by modifying the DNA sequence. And this relationship between the DNA sequence and the properties that are associated with these changes, it's called the genotype to phenotype relationship. So you can kind of imagine this on a hyperdimensional kind of landscape where you have different changes and variations in DNA sequences and what their impacts on cellular properties are. And so on a fundamental level, that's what we're trying to address, right? We're trying to understand or map out this genotype to phenotype landscape. And the work so my lab mainly works with microbial systems, right? Because microbes can grow really fast and we can actually culture them to very high densities in the laboratory, what we're doing in my lab is to take advantage of this ability of biological systems to adapt and evolve in order to help us better understand how they work and help us map out this genotype to phenotype relationship. As far as the department goes, I love that we have a very collegial environment in the department, and that I think most faculty here have very good relationship with the students. I personally feel like I work better in an environment where it's friendly rather than competitive, and Texas A&M is it has that very collaborative culture, which I really like.